learn things, we researchers learn things that can be then useful for other researchers that work in other completely unrelated diseases like, I don't know, cancer, diabetes, whatever.
Fellow Homo sapiens. Now, the epilepsies can be simply horrific. But what can help some of us to keep going is the realistic hope and excitement brought on by neuroscience, genetics, biomedical engineering, and actually so many other subjects combined. Today, hear Jordi Duran from IQS Barcelona explain to us his exciting research into the ultra-rare epilepsy Lafora disease, the challenges he faces, plus how funding for research into Lafora disease can actually lead to discoveries that benefit those affected by other diseases too, like Alzheimer's and Parkinson's disease.
I'm Dr. Jordi Duran, and I'm from Barcelona, and I'm a researcher, I'm a scientist, so I do biomedical research on how the brain works and on conditions in which the brain deteriorates, especially in rare diseases.
Okay, and so it makes complete sense, I guess, then, that you have an interest in the rare epilepsies.
Yes, especially because among these diseases, maybe the one that I study with more interest is Lafora disease, which is a rare condition that is characterized by the presence of severe epileptic phenotypes. So yeah, that's quite what connects me to research on epilepsy.
And how rare is Lafora disease?
Lafora disease is a rare disease. It's what it's called an ultra rare disease. So very few people in the world have this disease, fortunately, because it is a very traumatic and severe disease. So I would say that maybe it affects. It's hard to estimate because since it's such a rare disease, it's hard to estimate. But the numbers would be every 1 people in a million. So
wow.
Yes, we think that now in Spain we have 8 to 10 cases, probably a few hundred in all the world. So it's an ultra rare disease. So you know that a rare disease is when it affects 2 people every 10,000. In Lafora, it's 1 per million, so it's an ultra rare disease.
And it's a disease based upon a genetic mutation.
Yeah, it is a genetic disease, as you were saying. So it's inherited. So it's a particular case of genetic disease because there are 2 genes that can be affected, and mutations in one or the other gave exactly the same disease. So yeah, one is called malin, the other one is called laforin. But mutations in these 2 genes give the exact same disease, which is Lafora disease.
From what little I know of this, it's a really, to be honest, well, epilepsies are never joyful, to put it politely anyway, but this is a pretty severe form of the epilepsies, and not just seizures, but other symptoms. Could you tell us about that, please?
I mean, the first dramatic thing about the disease is that it affects children. So the onset is normally if people are around 14, 15 years old. And then the thing is that epileptic seizures is only the first symptom. So normally the disease starts by epileptic seizures, but then the disease progresses very rapidly. And then these kids start having all sorts of neurological problems, and eventually they stop being able to talk, stop being able to walk, and become bedridden. And all the time they have these long epileptic seizures. At the end, they become in a status epilepticus, so they are almost seizing all the time. So it is a very dramatic and aggressive disease. And yeah, unfortunately, there is no treatment for it. So these kids that suffer the disease normally die 5 to 10 years after the first symptoms. So yeah, it is a terrible disease.
So it's basically a death sentence upon diagnosis, correct?
Yes, so far it is.
So far, yeah. And I think I read also that the symptoms other than seizures tend to be or resemble dementia.
Yeah, I mean, the brains of these patients deteriorate very fast. So not only because of the seizures that also affect the brain and can, deteriorate it, but also this, as I was saying, these kids suffer from dementia, as you were saying, from progressive neurodegeneration. So at some point they undergo dementia. So they have what is called child dementia. Of course, when we think about dementia, we think about old people. But in this case, there are also kids that have dementia because they have this disease or others. That so yeah, it is what they undergo dementia. So the brain progressively deteriorates and they lose functions. Every time they lose more and more functions. And as I was saying, at some point, eventually they stop being able to do normal things like talking, walking, and at the end they become bedridden, and still they have these dramatic seizures all the time. So yeah, again, it's a very aggressive disease.
But I think there is a slight, if one could say, a slightly positive thing to say in this episode, and that is you've been involved for quite a while now in research regarding this rare epilepsy. Could you tell us about that, please?
Yeah, we and other groups have been studying the disease for some years now. And when we started working on Lafora disease, we knew nothing. There was nothing about the disease. So the disease had been discovered at the beginning of the last century by Spanish doctor Santiago Rodriguez Lafora. That's why it's called Lafora disease. So Lafora identified that there were kids that were dying with this because of this disease. But the reason why these kids had this neurological condition, it was not clear. So some years ago, we and other groups started trying to understand what was the disease, what is the cause of the disease, and we and other groups could identify the problem. What is the problem in Lafora disease? And the problem is that the brain accumulates a substance that is called glycogen, that is a normal component of the brain, but when it is accumulated in excess, these accumulations make the brain deteriorate. So with this, again, not only my group but other groups found like a target. When you have a disease, you need a target that you can treat, right? You need a drug, what we call druggable target. You need to find a target, and then you need to find a drug for the target. So some years ago, we identified the problem, and since then we have been trying to find a solution for the problem. But at least we can say that in a few years we have advanced very much in the knowledge of the disease. So we went from knowing absolutely nothing to knowing what to treat, and now we are trying to generate actual treatments for the disease.
And could you give us an example of where you are in identifying treatments, and what stage are you at?
So, for instance, what we identified is that the problem is that the brains of these patients accumulate a lot of this molecule called glycogen. So we understood that if we could get rid of this glycogen, the disease would improve. At least that's what we were expecting, and we actually demonstrated that this is the case. So another group took advantage of a drug that is already available for a different disease that is called Pompe disease, and checked if this drug could also be beneficial for patients with Lafora disease. They did that on animal models, and it worked. Now, actually, the first patient in the world is being treated with this drug. The problem is that this drug is not specifically designed for Lafora disease, so it has. We still don't know to what extent it will be effective, but at least now the first patient in the world is actually being treated for Lafora disease with a drug that is what is called repurposed. So the drug was aimed at another disease, but now doctors are checking if this drug will also be beneficial for a Lafora disease patient.
How far through that are we in terms of this drug?
Again, this patient has been in the drug for several months now, and it's hard to say, but it looks like it is working. So the problem with this drug is that it's not designed for the brain. So when we want to treat the brain, there is an extra. So treating any tissue or organ is difficult, and designing drugs to treat diseases is always difficult. But the brain is especially difficult because we have a brain. Between the brain and the rest of the body, you have the blood-brain barrier. So the drugs that you design for the muscle maybe will not go into the brain because there is this barrier. So this is probably the problem with this drug, that it probably won't cross the blood-brain barrier very easily. So, but still, it seems that some is crossing, and that there is an amelioration of the disease in this patient. It looks like it's still soon to know. Also, the fact that it's only one patient being treated makes also the comparison very difficult. You don't have other patients to compare how fast they evolve. But the results seem to be promising. And talking to her mother, she's quite confident that the treatment is working. So yeah, we'll see. And now, of course, we are trying to find ways to make this drug more accessible to the brain. So, and to make it reach the brain better, so that it will work better. It will fight this glycogen better.
And in terms of all of this work that you've been doing, I understand that you've discovered things that could be beneficial for use of treatment in other diseases. Is that right?
Yeah, it is right in the sense that when it's important to understand that when you study a particular disease, you establish knowledge, and you get knowledge and pathologic mechanisms that can maybe be applied to other diseases. So what I mean is that by studying one disease, not only you benefit for the treatment of that particular disease, but for other diseases. And studying Lafora disease, we have learned, for instance, that accumulating glycogen is bad for the brain. So and we have found other conditions in which the brain accumulates more glycogen than expected. So, for instance, we have recently published a paper in which we show that, again, in animal models, that for instance, another neurodegenerative condition, ALS, amyotrophic lateral sclerosis. So there is also accumulation of glycogen in the brain and spinal cords of ALS animal models. If we prevent that, the disease progresses more slowly. So it seems that this pathologic mechanism that we have found for Lafora disease could also play a role in other conditions. So could also be, a target that we could try to drug in other conditions like ALS, but also in conditions like Alzheimer's disease or Parkinson's disease. So in many other neurodegenerative conditions, glycogen might also play a role. So the drugs that we're designing for Lafora might be also useful to, slower the progression of other neurodegenerative conditions like ALS, Alzheimer's, etc.
Amazing.
So yeah, and also by understanding what happens in Lafora and how this accumulation of glycogen induces epilepsy, we could also learn from what is epilepsy and from what are the mechanisms that drive epilepsy in other diseases. So, but I would say is not something particular to the research on Lafora disease. So all the research that we do in different diseases are connected, and we learn things, we researchers learn things that can be then useful for other researchers that work in other completely unrelated diseases like, cancer, diabetes, whatever, So research is always connected and we learn from each other a lot.
And this is a wonderful example, in my opinion, of why clinicians and researchers who don't necessarily right now focus on the epilepsies should actually be interested in epilepsy research. And of course, in this particular case, Lafora disease. And wow, I just think it's really exciting. A huge thank you to Jordi for sharing with us the exciting work that he and his team are doing behind the scenes, providing those affected by the currently fatal ultra-rare genetic epilepsy, Lafora disease, realistic hope. Do check out the charity Chelsea's Hope if you'd like to learn more about Lafora disease. Plus, there are links to Jordi's work on the website torierobinson.com.